These are in a PowerPoint. I mentioned this, everybody might not have been back from break. I will share these PowerPoints once we go through them as a reply to the announcement. And then I also try to share a video for each one. Sometimes I might not get to make a video for them, but you'll at least have the PowerPoints where I include, there's actually information from your PowerPoint slides that I sprinkle into this so that we go through the information and you're actually learning as we're going through it. Similar to the Kahoot, but this one gives me a little bit more leeway to give more instruction between. Okay, so if you're on pollav.com/DocJulie, or if you're texting, you should be able to see something that says choose the correct response, and all you're going to have to choose is A, B, C, or D. I'm actually going to look at the responses on my phone so that I don't have to go back and forth off the screen on my computer. All right, somebody chose A. I haven't shown you the question yet. That's okay. It's good that I know it's working. I know that somebody's on there. Okay, first question is, what is the function of the diaphragm on the compound microscope? Somebody has a question, so now I have to look at this real quick. Yes, it's like a quiz. I have questions and then I have information that we cover between each one that explains the information if you didn't know the answer yet, which is likely the case because this is how we're covering the material. Like I mentioned, I will also have content videos where I just go through the information for the chapter in those need to know documents. So it's kind of our way of repeating the information. We're doing the quizzes and then I do the explanation in the quizzes, and then I also will share a video that is what I call a content video. Those will be underneath the modules, underneath the sections where the PowerPoints are and everything. All right, really different answers. Everybody said A, B, C, and D. Most were for C and D, but the correct answer is controls the amount of light entering the condenser. There is a picture in your PowerPoint, I think, and in your textbook that explains this, but I found this one. If I use a source that is from other than your textbook, I will include that URL for that source at the bottom. That's true for the need to know documents and for any of the quiz questions and everything that I have. But so this one is a nice little guide that just tells you what each part is for. And I'm trying to think. I think it was for the video that I created today, the chapter 3 content video. I mentioned this in there and we talk about how to calculate magnification. Now I'm saying the same information to too many students. And now I'm starting to forget what I already told you guys and what I did not tell you yet. Okay, moving on to the next one. The ocular lens is used to do what? Which of these is the ocular lens, also known as the eyepiece? Somebody put the answer in the chat. It's okay. No, that you enter it if you didn't. Maybe you weren't here when we did it. We're entering it on pollav.com. So if you're doing it on the internet, you can go to pollav.com/DocJulie. Or if you want to use your phone and respond via text, you text DOCJULIE, no spaces, to the number 37607 to join. And then after that, all you'll have to do is choose A, B, C, or D. You could do it in the chat, but it just makes me have to go back and forth. It's easier this because I can actually see a graph. I don't know if y'all can see that. You probably can't see it, but it shows me a graph of what the students are voting for. The correct answer is to remagnify the image formed by the objective lens. The objective lens is the major amount of magnification. So those are usually 10x, 5x, 20x, 40x. Because there's two lenses, it has to actually flip the image. So the ocular lens is the eyepiece, the one that you actually look through. Let me go back to the microphone picture here. This is the ocular or the eyepiece. That is usually 10 times magnification. And then the objective lenses are the ones that can go to higher magnification. And the way that, oh, no, that's right. I thought I went to the wrong question. The way that you calculate the total magnification is the objective lens, which is the 10 times magnification. Oh, I'm keep saying it backwards. The objective lens is the one that's different. So that's the 10, 20, 40, or 100. The ocular lens is 10 times. So the way that you calculate total magnification is the objective lens times the ocular lens. I think that's a later question, but now I just gave you a heads up for it. All right. 57% of you said B. The measure of the light bending ability of a medium is which of these? We have votes for A, B, and C. I'm going to go ahead and show the response, which is C. It is refractive index. Refractive index is the bending of the light. So light will naturally bend when it approaches a substance. And the important thing that you need to know about this, besides the fact that the refractive index is the bending of the light, that's the definition, is that at the 100x objective, which is the oil immersion objective, that's the highest magnification that a light microscope will go to. You must use oil immersion to bend that light so that it focuses it through the objective. Otherwise, the image will appear fuzzy and have poor resolution. That's fine if you can't do it on there. You can still do it on the chat if you want. Okay, next question. Oh, here's a picture that's showing you this. This is in the need to know document also. Most ocular lenses, see a lot of these I already gave you a hint for. Most ocular lenses magnify specimens by a factor of which of these? The ocular lens is the eyepiece. Most of you are voting for A, which is good. That means you were listening. Some of you voted for others. 10x is the most common eyepiece or ocular magnification. Okay, I don't have a description for that one. The ability of the lens of a microscope to distinguish fine detail and structure is referred to as which of these? Whoops, I forgot to clear responses. No, it's not responding. Sorry. Resolution is winning. Magnification is coming in a close second. The answer is resolution. There's two parts to the image that we look at through the microscope. The first part is the magnification, which is how many times larger than the naked eye you're seeing the object. That's what the 10x means. It means the object, when you look through the microscope, is 10 times larger than it would appear looking at it by the naked eye. So the total, why won't this let me? It's not letting me clear my responses. I'm getting frustrated. Of course, something's going to mess up. Everything can't work. Oh my gosh. Okay. I'm going to have to pull it back up again. So that's the magnification. That's how much larger the image is than looking at it with the naked eye. Resolution is how clear is that image? How well are you able to distinguish 2 points? So you can look at it under the magnification, but until you put it in focus, the resolution will not allow you to see that image. This is just totally not letting me clear the response. So it's kind of a moot point right now. Oh, it's not good. I guess it's just messing up for everybody. Yay. All right, well, let's just finish getting through this. So now I'm not going to be able to see your responses, so you don't necessarily have to respond. But I'm just going to walk through each one of these. So next one is which microscope uses an opaque disc to block light that would enter the objective lens directly. This next section gets to the different types of microscopes. And really what you're going to need to know for these are the information that is in this clicker PowerPoint. These would be the types of questions that I'm going to ask about these. The answer is dark field. There are different types of light and different angles of light that are illuminated towards the specimen that allow these different types of microscopes to let us see specimens of different types that may be more challenging to view with a certain type of microscope or staining. Okay. There is a table in the Chapter 3 Need to Know PDF, which I will be posting as soon as we finish. Actually, I think it might be under modules already. That has a nice table that summarizes the different microscopes and how they work differently, all of the different ones. And then I also cover that in the Chapter 3 video that I'm going to post. Okay, moving right along. Which microscope uses ultraviolet light as the light source? See, now it's doing some. I don't even know what's going on. Somebody says fluorescent microscope. That is correct. So fluorescent microscope is the one that uses UV light as the light source. And then there are fluorescent probes that are attached to something, usually an antibody, that binds specifically to a protein or a structure in the cell, like a key fitting into a lock, that allows us to visualize it. And the fluorescent probe will reflect off a color of light once the UV light has hit it. And this picture on the right kind of just shows you what an example of a fluorescent microscope would show on the image. I actually used to do research in a neurobiology lab where I used a fluorescent microscope. And you have to turn off the lights in the room so that you can see the light that is illuminating from the specimen. Next one. Which microscope illuminates specimens with blue light and produces 3D images? I'm going to go ahead and pull up the answer because I don't know if people are voting or not anyways. It won't let me clear. That is a confocal microscope. So like I said, different types of light are illuminating the specimen at different angles, and that gives us different views. It is much clearer picture, so a better resolution using the confocal microscope. So there's different benefits to using different types of microscope. Here's one with a fluorescence in addition to confocal, so some of these will overlap. But you can really see on a specimen where the different substances are situated in the cell, which is helpful. Okay, next one is which microscope forms an image from two sets of light rays, one from the light source and the other from diffracted from a structure in the specimen? That was a complicated one. Two sets of light rays. That is phase contrast. So that is hitting it at different angles, and you're going to have shade that forms at different angles, and that allows for you to see the specimen in different. One of the other benefits of a phase contrast microscope is that we can examine living organisms. Many times we have to fix an organism to the slide, which we usually do by lightly heating it, and that causes it to stick to the glass. And then we can look at the sample, but the problem is that it kills the organism. So you can't look at a live organism with many different types of staining and microscope types. All right, next one is which microscope is especially useful in studying the surface structures of intact cells and viruses? So the viruses part is kind of a hint there, would be one of the microscopes that is the highest amount of magnification. Answer to this one is scanning electron microscope. So if you chose that, good job. Scanning electron microscope provides really cool 3D images. It doesn't say cool in here, but they're really cool. Any of the pictures that you look at that look like a really, really tiny, cool image of a 3D structure is scanning electron microscope. So it scans the surface and shows the surface structures. Which microscope is extremely valuable for examining different layers of specimens? And this is up to 100,000 times. Answer for this one is transmission electron microscope. In transmission electron microscope, we take slices, transverse slices of the specimen, and we'll actually see internal images. Transmission and scanning electron microscopes both have a really high magnification. I've seen some that say they go all the way up to 500,000 times magnification. A light microscope generally only magnifies about to 1,000 times maximum magnification. Okay, this is just some more information in this. So the difference between transmission and electron would be that transmission electron microscopy is going to show the internal structures of a specimen, whereas the scanning SEM is going to show the surface, the 3D surface structure. All right, kind of gave you a hint on this one. With us, and we often abbreviate these SEM, so I'm going to start doing that. And SEM objects are generally magnified. We got all kinds of votes going on. 1,000 to 500,000 times, because there can be a huge range of magnification, but all electron microscopes magnify to the highest degree, which is up to 500,000. I've seen some that even say up to 1,000,000 times. All right, before microorganisms are stained, most appear blank when viewed with a bright field microscope. This would be an interesting one. I keep forgetting that I don't know if it's clearing or not. Sometimes it's clearing, sometimes it's not. That is colorless. So the reason that we have all of these different staining techniques that you're going to learn in chapter 3 is because of the fact that cells are clear, and when you look at them through the microscope, you can't see anything unless you stain them. So these different types of stains stain different structures and allow us to visualize the organisms. This next section, we're going to learn about the different types of stains and what the uses are, what we use to see. Yeah, one of the big techniques that you're going to learn is the gram staining, which is kind of fun to do in the lab, and I'm sad that we don't get to do it, but you will get to watch videos about it. So a simple stain is when you just apply a single dye, and basic dyes are going to be the ones that are usually used to stain cells because they are positively charged and they attract to the negative charges on the cell membrane or in the structures. Oh man, I probably just gave you the answer to this question. The chromophore, which is the part of it has the color, chromo means color, of basic dyes is blank. Is it a cation or an anion? And is going to be attracted or not attracted to most types of bacteria. Basic dyes are cation. I mentioned that the cell membrane and many structures are negatively charged, and so it is going to attract to most types of bacteria, and for that matter, most of our cells as well, eukaryotic cells. Okay, next one. Preparing smears for, oh, this is just more information. So this just breaks down, this is actually straight from your actual PowerPoint, and I have it included in the chapter 3 need to know document. But basic dye, the chromophore is a cation. Acidic dye, the chromophore is an anion. That is going to allow for the staining of the background instead of the cell, an acidic dye. That is known as negative staining. Somebody have it. Oh, somebody needs to mute. One moment, please, while I figure out who's not muted and mute them. Okay, I think I got it. Okay, other thing you need to know about the staining techniques is there's 3 basic types. There's simple staining, which is just using one stain to stain a structure. There's differential staining, which, as the name suggests, allows differentiation of different types of organisms or different structures by showing different colors. And then there's special staining, which is certain techniques that are used for viewing types of cells or organisms or structures that will not stain readily by different staining mechanisms. All right, next, which one of the following is not a kind of staining technique? I just told you all 3 of the ones that are. So the answer is selective. We'll talk later, that's about a type of growth medium where you select for the growth of a certain organism. But these first 3 are the basic staining techniques. I mentioned this one also. The process of fixing a slide attaches the microorganisms while simultaneously, the downside of these is that it kills the organism. So all it does is basically kind of glue the organism to the slide so that when you stain it, it doesn't wash off. Downside, it kills it. I mentioned we do this normally by heating. Boy, this is a big old PowerPoint. I didn't remember having this many. All right, a technique used for preparing a slide with colorless bacteria against a colored background. Nobody voted on it. It is a negative stain. That is the acidic dye that is negatively charged and it's repelled by the cell membrane with all the negative charges on it, and it actually will stain the background. And it will be basically looking like a white looking structure that's illuminating light on a dark background. Okay, getting into the gram stain technique. There's several things you're going to need to know about the gram staining. The steps of the process, what order the steps go in for the different staining steps, and then the function of each one of these, and then what color the gram positive and gram negative bacteria will be after the staining. So mordant in the gram stain is, uh-oh, my answer slide went bye-bye. See if I have it here. Okay, it just got mixed up. Is iodine. A mordant is a substance that basically glues the dye to the organism. Let me see real quick. Somebody had a question in here. Oh, my chat's gone. Oh, some it's letting you answer, some it's not. I'm looking at it because there's still some stuff coming in, but in these PowerPoints, I should mention, and in the need to know documents, sometimes I will have additional videos that are linked in there that help to explain the material. So if you see something that has a hyperlink, it's a video that I added in there to give you more information. One of the things we're going to learn about in chapter 4, which we're going to do chapters 4 and 5 on lab days. When we have 4 chapters in 1 week, I like to do 2 of them on lecture day and 2 of them on lab day because our labs are short. But gram staining is used to differentiate between 2 major groups of bacteria. And these are different by the structure of their cell wall, which we'll learn more detail on that in the next chapter. But gram positive have a thicker peptidoglycan layer, which will stain purple. And then gram negative have a thinner peptidoglycan layer and a lipid outer layer, and those will stain pink. These are the steps in the gram staining. It's crystal violet followed by the iodine, which glues the crystal violet to the cells. Then we apply a decolorizer, which can be either alcohol or acetone, and that will wash the crystal violet, which is purple, off the gram negative bacteria. And then we use a counter stain, which is the safranin, that will then stain the gram negative bacteria pink. I might have other questions on the gram staining that I just answered. Flagella. The staining for flagella is structures that are difficult to stain with traditional stains, and that would be a special stain, like I was talking about. And some of these are more than one type, but the stain is carbol fuchsin. If you look on the, there can be other stains too, but that's the main one. If you look on the need to know document table that I have on there, it actually has these all in a table. But in your PowerPoint, it just kind of has them as information. Flagella are structures that are tail-like structures that allow for movement, and this just reiterates that uses it. This is showing you a spiral or spirillum bacteria, and it has the flagella at the end. Capsules are stained using I gotta move this so I can access it. Oh. Now my slide's not responding. That is a negative stain. Capsules are made from a glycocalyx, which is a carbohydrate substance some bacteria release, which is this kind of sticky substance that protects it from the immune system. And these sticky substances resist staining. And so we have to use a negative stain to stain the background, and then the capsule will just reflect off light and appear white. I keep forgetting to show the information while I'm talking about it, but this is basically everything that I just said. So there's several different types of negative stain that can stain the background. One example is India ink or nigrosin, and they would allow it to eliminate the capsule, which kind of looks like this. These are all different techniques that are used in a lab. Say, for example, somebody comes into the doctor, has an infection. We are going to take a sample of the infection, and then we are going to send that off to the lab, and they're going to do these different staining techniques and these use these different microscopes in order to identify what species the infection is being caused by, so that we can target the medicine to be able to treat that specific infection. I don't know what's going on here. It's starting to act funny. Maybe I should stop recording so it doesn't go bye-bye. Okay, I'm just going to try to hurry up and finish. Endospores are stained using Schaefer-Fulton stain, which is also known as malachite green. Endospores are structures, which we'll talk about more later, where a bacteria is in a harsh environment and it almost like a seed for a plant. It is a protective structure that keeps it from drying out, and they are very resistant to staining as well. Why is my thing not clicking? I don't know what's going on. My computer's starting to get upset. No, hang in there. We're almost done, please. Okay, here. I have the video for endospores linked there that gives you more information about that. Oh my gosh, now I'm completely on the wrong slide. I don't even know where I was. Here it is. Okay, so that's the information that I was talking about with the endospores. They are counterstained with safranin. And so when you have an endospore staining, it will look like this. The green is the spore part of it. That is stained with malachite green. And then the rest of the cell will stain with the safranin, and it appears pink. What color are gram-negative bacteria at the completion of the gram staining process? That is pink. If you chose pink, you are correct. So in this picture, you have some gram-positive bacilli. These are rod-shaped bacteria. And then the gram-negative bacteria also look like they're rod-shaped. They might be coccobacillus. I like to call it tater tots. They kind of look like a squished rod. But that is one of the things that you need to know for this chapter. Gram-positive stains purple. Gram-negative are pink. Appearance of Mycobacterium leprae after they are stained with an acid-fast stain. This is another different type of bacteria that has a different substance on the outer surface of its cell wall that also resists staining. So acid-fast stain is another type of special stain. And these would appear red. Two examples of acid-fast bacteria are Mycobacterium and Nocardia. These are, as we go along through the session, you're going to start to learn the different species names of the bacteria, but they have a waxy substance on the outside that actually resists staining. And then this last slide just gives you a chart that explains the different steps of the acid-fast staining. I'm going to stop the recording, if it's recording. I don't know if it is. This is for my Loom, not for my Skype recording. Yeah, see, it's not working. I thought it wasn't going to work. I could tell by the way it was acting silly. Okay, so that is the chapter 3 information. What I'm going to do is we are actually going to stop now, and I am going to share the chapter 1 recording, so you can go ahead and watch that right now if you have time, because technically we still have an hour left. And then I will also share this PowerPoint that we just went through and the video recording of it. Well, actually, I won't share it. I can only share it through Skype because the Skype recording. Let's see if the Skype recorded it. I was trying to do it on Loom so I would have a URL to share, but it's not working.